Okay, can somebody confirm you can hear me?
I can hear you.
Great. It's giving me a note here that your speaker is not working properly. That's why I was asking. I'm not sure what's going on, but if you can hear me, everything's fine. All right, let's begin. My computer is slow because it's still processing the lecture. Let me close that down. Come on. All right, let me share my screen here. Which one of these is it? It's got to be that one. Nope. There we go. So we're going to be discussing today Lab 5, Streaking for Isolation and Culture Media. Please read the unknown project instruction files for next week. I will discuss the unknown project next week. You can find the unknown project instructions in Canvas, which I'll show you in just a minute. Oh, good. My lecture page just finished, and so my computer will start working normally now. So scroll down to week 5 lab, and the unknown instructions.docx is the file you need to read. Any question about any of that? All right, so today we're doing lab 5. Yep, get it in time. So let's go to lab 5. Lab 5 is the cultivation of bacteria, culture media, inoculation, and isolation. There is a little bit of reading. What is that? About 4 pages there on culture media and just a page on obtaining pure cultures. There is some video clips to this lab. There is no virtual lab. Upon successfully completing this lab, you should be able to, one, understand the importance of proper specimen collection and transport to the lab. Two, be able to explain the 5 principal steps used by microbiologists to process specimens. 3, be able to differentiate between various types of culture media. 4, explain the purpose of streak plate culture technique, aka streaking for colony isolation. So know what that is and how to do it and what the purpose of it is. And then 5, define the following terms: selective media, differential media, and reducing media. Is there any question about what we're going to be doing today? All right. So the ability to isolate pathogens from a clinical specimen submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, they'll dry out, and some are sensitive to exposure to oxygen, almost all anaerobes. And these require a special transport medium. Once a specimen is received in the lab for microbiological culture, microbiologists employ the 5 principal I steps to process it. The five are inoculation, isolation, incubation, inspection, and identification. Inoculation is the placing of the specimen on an artificial culture medium to initiate its growth. Isolation is the spatially separating out microbial growth into distinct and pure colonies. Incubation is the placing of the culture medium in an environment under conditions to facilitate its growth. 4, inspection is examining the culture for the presence of microbial growth. And then lastly, there's identification, performing additional specific tests to ID any pathogenic microbes. So even though you might isolate it, your work on identifying it has just begun. Any question about any of that? The 5 steps will be described in detail after first discussing different types of culture media. So let's talk about the different types of culture media. There are selective, differential, reducing culture media, and then the one we've already talked about, is a general purpose culture media. You should be familiar with general purpose media, and that is Na for nutrient agar, NA or NAYE for nutrient agar with yeast extracts added, and then TSA or tryptic soy agar. All of these are general purpose media that have no selective capability, and we just employ them to grow microbes cheaply and easily. So these plates are both advantageous in that they support the growth of many different cells, and they are cheap. To promote the growth of a fastidious microbe, these microbes will not grow on a general purpose media. They have very specific nutritional requirements, and we use enriched general purpose media. For example, we can add 5% sheep's blood to the growth media to help the organism grow. And blood agar plate, also called BAP, is an enriched general purpose media for getting many microbes to grow that would not grow on like TSA, for example. And all blood agar is TSA with 5% blood cells added to it. And then chocolate agar is another enriched general purpose media. Any question about that? So we have general purpose media, enriched general purpose media, and then we have selective media. These media contain chemicals that inhibit or halt the growth of some organisms. When you're using selective media, keep in mind 2 things. First, a selective media isn't necessarily perfectly selective, meaning given enough time, other organisms can grow in it, even though they should not, or meaning it selects against it, but with enough time, you can get the organisms to grow that you're trying to select again. Second, even those organisms that are supposed to grow may form smaller colonies and may grow more slowly on the selective media than they would on a general purpose media. There are different kinds of selective media, and we'll talk about a few of them. There's also differential media, and these media contain chemicals that produce visible differences among different groups of organisms when they are grown on that media. These differences are unrelated to how well the organism can grow on the media. Differential media allow dissimilar bacteria to be visually distinguished from each other. Any question about that? Lastly, there's reducing media, And these media promote the growth of anaerobic bacteria because these cells require either low or no oxygen conditions in order to get these organisms to grow. It's important to note that a culture medium can be classified into more than one category. For example, McConkey's agar is both a selective media and a differential media. Blood agar is also both a differential media and an enriched general purpose media. Any question about any of that? So here we're looking at McConkey's agar, and it is selective. If you get growth on McConkey's agar, it is almost always gram-negative. Usually McConkey's agar is also almost 100% selective, meaning you either get growth or you don't. There are some selective media where you can get growth, but usually it's less than the normal growth. Anyways, McConkey's agar, since we're getting growth on McConkey's, both of these are gram-negative. And you can see differences between these two species on the McConkey's agar, and that's because McConkey's agar is differential for the fermentation of the sugar lactose. And if you see the sugar lactose being fermented, you'll see pink colonies as well as pink around the growth. And that's because the fermentation of the sugar lactose produces acids which cause the medium, as well as the growth on the media, to turn pink. On the other hand, if the bacteria cannot ferment the sugar lactose, then they will be their normal color. And in this case, it is either white or off-white. And that's the normal color of this bacteria that cannot ferment the sugar lactose. Any question about any of that? All right. Blood agar is an enriched media, as I mentioned, allowing you to grow fastidious organisms. On blood agar, it is also differential in that you can look at the differential hemolytic activity of the bacteria. You can have 3 different hemolytic activities of bacteria. You can have gamma hemolysis, shown here, and that's actually a Greek gamma, and the red blood cells are red all the way up to the off-white bacteria, meaning there's no hemolysis, and that is called gamma hemolysis. There's also alpha hemolysis, and let me blow that up a little. Alpha hemolysis is partial hemolysis, and you can see the red is red up until this dark or greenish ring. It's usually a dark or greenish ring. This case is dark, which is shown around the alpha. The alpha is the whitish or off-white bacteria growing, and that dark ring is the partial hemolysis of the red blood cells that appear dark. You can see inside the alpha, that region is totally dark. Any questions about alpha hemolytic activity? The final hemolytic activity is beta hemolysis, and the white bacteria in the shape of a beta, that's a Greek alphabet, by the way, Greek letter. The white is the bacteria, and around the white, we see this clear region there that is the complete hemolysis of the red blood cells. And so that is beta hemolysis. You'll notice a dark ring around the beta hemolysis, and that's actually alpha hemolysis. It is normal to see alpha hemolysis surrounding beta hemolysis. Any question about any of that? So this table is showing you the different types of media: general purpose media, tryptic soy agar, nutrient agar with yeast extracts, differential and enriched media. There's only two that we're talking about: blood agar, and then excuse me, there's only one differential enriched media, and that's blood agar. The other enriched media is chocolate agar, which is CAP. Chocolate agar is not differential. It is only an enriched media. And how it differs from blood agar is you add the blood as soon as the agar comes out of the autoclave, and that agar is too hot to retain the blood cells, and they lyse, giving the appearance of the agar a brown color, and it looks something like chocolate, why it's called chocolate agar, but in reality it's lysed red blood cells. Any questions about chocolate agar or blood agar? We also have some selective and differential media. They are both selective and differential, and that would be MacConkey's or MAC, or eosine methylene blue or EMB, or Hectone agar, HE agar. And they are both selective, or all of them are selective, that if you get growth on these plates, it's most likely to be gram-negative because the gram-positive cells are selected against. They're also differential, and they are all differential on the basis of their ability to ferment lactose. If they can ferment lactose, they will be a different color. MacConkey's agar is pink. EMB agar is a dark color or a greenish color. Hectone agar, if can ferment lactose, it'll be salmon colored. Any questions about any of that? We also have selective media, and there's only one that's only selective you need to know about, and that's phenyl ethyl alcohol, or PEA. It is selective and selects against gram-negative cells. So on this plate, gram-positive cells will grow. And then there's mannitol salt agar, which we may have talked about briefly. It is both selective and differential. It is selective because it has 7% sodium chloride in it, and only bacteria that can tolerate 7% sodium chloride can grow on mannitol salt agar. Most bacteria cannot grow above 2% sodium chloride. So if it can grow on mannitol salt agar, that means the bacteria like growing in a high salt concentration, 7% sodium chloride. It is also differential, and it's differential on the ability to ferment the sugar mannitol. If the bacteria can ferment the sugar mannitol and it can grow on the mannitol salt agar plate, it will have a yellow color around the bacteria. So we call it a yellow aura around the colony of bacteria. If it cannot ferment the sugar mannitol, it will not be yellow and will be the normal color of the mannitol salt agar, which is kind of reddish. Now, with the selective and differential media, you can only do the differentiation if the bacteria can grow on the plate. If the bacteria cannot grow on the plate, you cannot say anything about the differential activity of the bacteria. Any question about any of that? All right, inoculation is just putting the bacteria on the plate. Isolation is isolating the different species of bacteria. In microbiology, we usually work with pure cultures because if you have a mixed culture, let's say 3 different species are in a tube, and then the tube turns yellow, you don't know which species or all 3 of them change the color of the tube to yellow. And so you wouldn't know if you're trying to study one of the species, if that species was positive or capable of turning the tube yellow. So we study a pure culture, and then if the tube turns yellow, we know that that pure culture caused the tube to turn yellow. Any question about that? Louis Pasteur was the first one who developed the concept of a pure culture, and he actually had a very labor-intensive way of obtaining a pure culture. What he did was he got a species, well, didn't get a species, got several, let's say he got a mixed culture of bacteria, got it to grow, made several different dilutions of that culture, and then replicated, plated like 10 times each dilution. So if you had 5 different dilutions, you would have to set up 50 different tubes. And if in that dilution, only one of the tubes got growth in it, and 9 of the other tubes did not have growth in it, Louis Pasteur correctly concluded that that one tube out of the 9, or the one tube out of the 10 that had growth and the other 9 did not have growth, was probably a pure culture. Any question about any of that? That was a very difficult way of obtaining a pure culture, and fortunately, Robert Koch developed a much easier way to get a pure culture. And what he did was he streaked out his bacteria on a solid media, getting isolated colonies, and then took cells from one isolated colony and streaked it out on a second dish to get isolated colonies again. And when he passed the cells through 2 colony formation events, he correctly concluded that the cells were most likely pure. And in over 95% of the cases, those colonies are pure, and you have a pure culture, which is good enough for working in microbiology. Now, let me point out that if you were to only do this one time, the chance that the colony is pure is only 70%, and that is not high enough for microbiology. Any question about any of that? All right, how do you get do the colony streaking is someplace when you're streaking out a plate, at some place you'll be streaking down isolated bacteria cells, which will then grow up into isolated colonies. And this figure, it's showing you that the isolated colonies are coming in the last quadrant, the 4th quadrant, but it doesn't matter if it happens in the 3rd quadrant or the 2nd quadrant or even the 1st quadrant. All you want is isolated colonies. And then you take from one isolated colony and then streak it out on another plate to get isolated colonies. And then you take the cells from this plate where the cells have gone through 2 isolating colony events, one here and one there. And then you can grow up those cells and you have an over 95% probability that those cells are pure. Any question about any of that? All right, so what you do is you get out your, pretend this is a plate, get out your loop, sterilize it, pick up your bacteria, and then you streak it in quadrant 1. And the highest concentration will be when you're first streaking, and as you go down, the dilution of the bacteria will decrease. You then take the loop and pass it in the flame or the Bacto incinerator to sterilize it. And then you come into sector 2 and you streak a little bit into sector 1. So sector 1 would be right here. Let me go ahead and show it with the mouse so you can see that a little better. So streak out sector 1 and then take your loop and sterilize it and then streak in sector 2. You should be starting in sector 2. This picture isn't quite good. And it shows that here you're getting only the diluted part of sector 1. You do not want to bring it way up here where the bacteria are highly concentrated in sector 1. You want to get it only in this portion where the bacteria are diluted in sector 1. And then streak out the bacteria to get sector 2. And then take your loop, get it in the Bacto incinerator, sterilize it. And here they got it shown right. You put it on sector 3 and then streak it into sector 2 to pick up the cells on the loop. Although you shouldn't come all the way over to here. You should only come where the bacteria is diluted in sector 2. So up to about here. And then streak it out. And it's only showing you a few streaks. You should be streaking it out about 20 times in each sector. You can streak over yourself. That's not a problem. But streak it out in sector 2. And then sterilize your loop. And actually, this one is much better. It's showing that they got the sector 4 bacteria from streaking on sector 3 where the bacteria is diluted. Doesn't show it where the bacteria is highly concentration right here. And then streak it out to get sector 4. And in this case, the isolated cells were put down in sector 4. So we got isolated colonies coming in sector 4. Now to get your pure culture, you take one of the isolated colonies and streak out another plate to get isolated colonies. All right, any question about that? All right, so in the steps for what a microbiologist does when they're trying to culture something that comes from a clinical specimen is they will put it in an incubator. That's called incubation, where you put the cells on a culture media to get the cells to grow. And then you put it in an environment to cultivate the microbes. In our microbiology lab, we have only simple incubators that only heat the culture to a certain temperature, and they add humidity, and then you incubate it for a certain amount of time. And that's all our simple incubators do. This incubator is more complex, and it can do all of those, meaning it heats the microbes while they're growing. It's putting humidity out into the microbes. And then this one, when you shut the door, it will have levers for putting out so much oxygen and so much CO2. So you can control the oxygen level as well as the CO2 level that the cells are exposed to. And then, of course, the time. You grow it for a certain amount of time. Any question about any of that? The humidity is actually important because when you're growing your microbes in the incubator, the liquid will come out of the plates, or in this case, the dishes, and will evaporate. And eventually that will kill the cells. Without adding humidity, the plates will dry out within about a week. And if you add humidity, all it is a container of water there, and then the heat simply brings the humidity out. You can have your plates in the incubator for up to 2 weeks. Any question about any of that? All right, you then inspect your plates to see the obvious visible colony morphological variations. You can usually see differences between different bacteria. That would be differences in the colony shapes and other colony descriptions, like the size, the shape, the margin, the elevation. We'll talk a little bit about that. But that would only begin to get you to ID the species. You next do your identification test, and that will be usually running the pure cultures on further biological tests to try and ID the species. Any question about any of that? All right, know the terms of this lab. There is some video clips. In the first video clip, you only need to watch it to 4 minutes and 15 seconds. It's going to be showing you the inoculation of an agar plate using street plate culture technique. There's only 3 sectors used in this video, and it doesn't matter how many sectors you have on the plate. Most people use either 3 sectors or 4. And then we have some videos on beta hemolysis, alpha hemolysis, and gamma hemolysis. So watch those videos. And then watch the video on McConkey's agar. You only need to watch it up until 3 minutes and 50 minutes of the video. And then we have a little video on mannitol salt agar. The virtual lab we have on practicing preparing street plate no longer works because it requires flash. So after that, we're going to be doing the lab. Remember to work in the lab worksheet because some of the questions may have changed, as well as there may have been added questions. So the worksheet for lab 5, first describe a natural circumstance where staphylococcal species, meaning a member of that family, a bacteria, how it might benefit from salt tolerance. So think of a place where Staphylococcus can grow, and it will benefit from growing because it's tolerant to salt. Has to be a natural circumstance. You can't tell me something like growing on 7% sodium chloride media, because that's not a natural circumstance. And then 2, how might hemolysis benefit a hemolytic bacteria? So this bacteria can lyse red blood cells. How may that benefit the bacteria? And then 3, imagine you have a culture containing a single bacterial species. This culture may be one of several possible species. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to identify the unknown bacterial species. You need to name 2 properties from each of these plates to help you identify the species. Any question about that? And then 4, what is the main goal of streaking for colony isolation? 5, what happens to the growth on the plate if you do everything correctly, but you forget to sterilize the loop in between sectors when you're streaking the plate for colony isolation? You need to be specific and discuss the growth on each sector. 6, what happens to the growth on the plate if you do everything correctly, but you transfer from stock culture to inoculate each of the sectors on the plate? Once again, you need to be specific and discuss the growth on each sector. Any question about that? 7. Suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species, meaning you streak the plate from a mixed culture. How can you use this plate to create a new pure culture of only one of the bacteria? Be specific in your answer. If you're not specific, I will count it wrong, or at least partly wrong. And then 8, in the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. Denote whether each medium is non-selective or general purpose media, selective media, or differential media by placing an X in the appropriate column or columns, meaning some of them you will put an X in more than one column. So nutrient agar, TSA, BAP or blood agar, CAP, McConkey's agar, EMB agar, HE agar, PEA agar, and MSA state whether it's non-selective, selective, or differential. And then 9, which agar plate or plates were inoculated without using streak plate technique? Plate A, plate B, or plate C? Any question about that? All right. It's only 7:38. I will be here until 9 o'clock to help you guys with the lab. Start working on the lab. Now remember, I will be here to help you for as long as there's someone in the lab. But once the last person logs out of the lab, I too will log out of the lab. Any questions? All right, go ahead and get started with the lab.